You know, The Little Mermaid taught me the virtues of living under the sea. And while Sebastian may have contended it's better down where it's wetter, I've always thought there was a point of diminishing returns right around when you see any fish that looks like this. Now for me, anywhere deep enough to see a freaky fish like that is more than deep enough. Anglers like that thrive in the bathypelagic zone, which starts 1,000 meters or more than half a mile undersea, and in the abyssopelagic zone 4 kilometers or up to 2.5 miles below sea level. But remarkably, the abyss isn't even the deepest part of the ocean. The Hadal zone, named for the underworld of Hades, describes ocean depths greater than 6 kilometers, extending down as far as 11 kilometers or almost 7 miles below sea level. These are mostly ocean trenches, which are in turn mostly created by the movement of tectonic plates, typically subduction zones, where 2 tectonic plates are smooshed together and one is driven beneath the other. Although these regions represent 41 to 45% of our ocean's depth range, the hadopelagic is less than 0.25% of our planet's sea floor, and while the Mariana Trench is easily the most famous example, there are 46 individual hadopelagic habitats in total, many of which formed about 65 million years ago. i.e. dinosaur times. So, this week's group of researchers asked in the journal Cell, what exactly are those habitats like? What kind of organisms live downstairs of even the freaky fish? These scientists hopped in a submarine about it. This is the Fendouge. Now sure, it's not the first crewed vehicle to dive down the Mariana Trench. That was the Trieste in 1960, the one with Jacques Picard, from the family of explorers for whom Star Trek's Jean-Luc Picard is named. But only a couple dozen people, including Titanic director James Cameron, have actually gone down there. So it's incredibly cool that we're sending these scientists. And they collected over 1,600 samples, some from the Mariana Trench, but also from the Yap Trench and Philippine Basin, gathering sediments from the full range of Hadal Zone depths. Now, people talk about how we know less about the depths of our oceans than we know about outer space. So let me take a moment to put these sample numbers in context. There have been a grand total of 9 previous studies like this. 9 studies is nothing in science. Pick any obscure topic you like, from snailfish to the way spaceflight gives astronauts a headache, and there will be more than 9 articles on that. These 9 studies span 34 Hadal Zone sites and gathered a total of 96 samples. 96. So, when this paper comes in with 1,648 sediment samples, it is truly a vast expansion of existing data about microbes at the bitty body of our ocean. And while those previous studies let us get a sample, if you will, of what the deep, deep, deep sea microbes are like, a study like this with tons of samples gives us enough to get a real look at the taxonomy of this ecosystem and, by examining their genetics, get a better idea of what adaptations allow them to thrive at unfathomable fathoms. No, but what we got? Well, a zillion different species for one. Our 1,600 samples yielded 7,564 species of archaea and bacteria, and almost 90% of them had never been seen before. That's not to say we've never seen anything like them before. When it comes to life, everything's related to something. A lot of the big-name bacterial phyla are also represented all the way at the bottom of the ocean. For instance, Pseudomonodota, the phylum that contains everything from Salmonella to the bacteria that causes cholera to the precursor bacteria to our own mitochondria, before their biological and technical distinctiveness became our own. There are also plenty of species from phyla that we don't see much here on land, but let me tell ya, Thermoproteota are big undersea. As the thermo might suggest, that phylum contains extremophiles who tolerate extreme heat like deep-sea vents. But just being in the same phylum doesn't necessarily make you closely related. This little skate, its name is little skate, I'm actually not nicknaming it, is in the same phylum as we are, but it's probably our most distant cousin within that vertebrate phylum. But because these prokaryotes are related, and because we have so many samples from such a range of prokaryotic phyla, we have the opportunity to see how this extreme environment shaped the ecosystem and genetics of these microorganisms. Now, some themes recur across these species. Anything that's down here has to have some way to deal with the wildly high pressure and the consequences thereof. So a lot of antioxidant adaptations to counter the generation of reactive oxygen species from the high pressure and near freezing temperatures. Also, a lot of what's called oligotrophy, the ability to thrive in low nutrient conditions. This is a type of what's called homogeneous selection, and it's probably the most classic evolutionary pressure you'd think of in an extreme environment. All the species down here have to have some way to survive the conditions, and that can create some evolutionary distance from shallow water species that don't experience that type of pressure. One of the microbial builds, if you will, down in the deep, runs a streamlined genome. Essential genes, including those adaptations I just mentioned. These are hyper-specialized min-maxers, and it fits with some previous studies that found that Colwellia bacteria from the Mariana Trench had a significantly smaller genome than their counterparts who were just from the normal deep sea. You might wonder why these species would have a smaller genome. Surely it's just strictly better to have more genes even if you don't use them all a lot? One reason goes back to just how nutrient poor the paedopelagic is. DNA is made up of elements like nitrogen and phosphorus, which could otherwise be used for growth and reproduction. The chemicals that make up our DNA usually seem like a rounding error to us, but this is an environment where every little bit really matters, especially to a single-celled organism. So, when by random mutation a gene is deleted from the genome in an individual, if that gene was important and conferred a meaningful advantage, that'd be a bad thing. But if you weren't really using that gene, there's actually a small advantage down here where every little bit helps, and that advantage creates some selective pressure to pare down to the essentials. Of course, the disadvantage of a small genome is that it's fragile. Maybe you don't need those genes right now, but if the environment changed even a little, those species are SOL. And in fact, our min-maxer is not the only type of build our researchers found in this marine microbial metagenome. Another strategy involved a much larger genome with more versatility, allowing them to thrive in a greater range of conditions, like in different sedimentary depths and in different oceanic current conditions. This is much more like what we see in a lot of our deep sea, but shallower deep sea bacteria, and our researchers suspect that these well-rounded builds may not be the product of long-term adaptation to the hadal biome. But that's a question for future studies. Frankly, these folks dove to the bottom of the ocean and expanded our existing data on the topic by multiple orders of magnitude. They've done a pretty fantastic job already. I'm sure they're thrilled, of course, but why do we care? Well, one, we can kvetch about how little we know about the deep sea, or we can just go ahead and study it, right? It's exactly what these scientists did, and as a result, we know way more about the prokaryotes of this biome, and those little buddies are the foundation of every ecosystem in the world. And ultimately, this teaches us more about how extreme environments shape evolution. We've studied organisms in all kinds of extreme environments, from those extremophiles in hot springs to, well, pretty much anywhere you could find a tardigrade. which for the record weren't a part of this study because they're animals, not prokaryotes, but for what it's worth, tardigrades can survive in the ocean trench too. But the more we study the hadal zone, the more opportunities we have to test and even challenge our hypotheses about evolutionary pressure in extreme ecosystems. Fortunately, the absolutely ginormous dataset has been made available to any scientist who wants to take a look. No submarine required. After all, that's got to be a lot of pressure.